So, welcome everyone, and thank you for taking the time to join us for today's webinar titled Advancing Cholangiocarcinoma Care in Canada Through Patient Voices, Research, and Advocacy. My name is Regis Fode. I'm Patient Affairs Manager at Servier, and it is my great honor to open this very special talk today. As many of you know, February is Cholangiocarcinoma Awareness Month, and this year, World Cholangiocarcinoma Awareness Day takes place this Thursday, February the 19th. This webinar is part of that broader effort to raise awareness around cholangiocarcinoma and to bring the community together around this important cause. At Servier, we're proud to support this webinar for the third year in collaboration with the Canadian Cholangiocarcinoma Collaborative, C3. And we strongly believe that progress in rare cancers like cholangiocarcinoma depends on collaboration across patients, advocates, clinicians, and researchers. And bringing these perspectives together helps amplify patient voices, deepen our understanding of the journey, and move us closer to better care and outcomes. And while meaningful progress has been made in recent years, important gaps still remain, and conversations like today's are key to continuing that momentum. So now it's my pleasure to pass it over to Leonard Angka, Project Manager at C3. Leonard has been absolutely instrumental in organizing this webinar. So Leonard, thank you so much for organizing this. And the floor is all yours for the opening remarks and to introduce the session's objectives and speakers.
Thank you so much, Thomas. So my name is Leonard, and really excited to introduce the speakers we have lined up for today's webinar. Every year, it's a very special time to that we get to host this and share with you all of the cutting advances in care and research, and also hear from patients themselves. So it's my pleasure to introduce Corri Desaulniers. So she was diagnosed with cholangiocarcinoma in 2022, but she's become a dedicated advocate and patient support for patient support and awareness. She's participated in many of our events and has been very active despite going through this disease and diagnosis. She's always kept such a positive attitude and a hopeful attitude and brings so much energy and good ideas to the community here in Canada. So very excited to hear from her. She's going to talk about her experiences with cholangiocarcinoma and also how that's led to her advocacy journey. Our next speaker after that is Dr. Robert Grant. He is a medical oncologist from Princess Margaret Cancer Centre in Toronto, and he specializes in biliary and pancreatic cancer treatment. His research applies artificial intelligence to clinical and genomic data to improve cancer outcomes. He also leads many different programs, including the Grayley Biliary Registry and the PanCureX Translational Research Initiative. He's going to speak to us today about biomarker testing or molecular testing and a lot more. After that, we're going to have Dr. Arndt Vogel. He is also a medical oncologist from the Princess Margaret Cancer Centre, and he specializes in translational and clinical research with a focus on hepatobiliary cancers and precision medicine. He's been a clinical investigator since 2006. He's led numerous clinical trials and coordinates the ESMO clinical guidelines for hepatocellular carcinoma and cholangiocarcinoma as well. And then lastly, we're going to hear from Dr. Rebecca Auer, who is surgical oncologist, also the CEO of the Ottawa Hospital Research Institute. And in addition, she co-founded the Canadian Cholangiocarcinoma Collaborative. She is also a caregiver to her husband, Adam, and she's going to lead the panel discussion that we're going to have.
Turn left onto Albert Street.
So I'm going to ask everyone to please mute their microphones once we get started. And then, yeah. So with that, I'm going to pass it to Corri to give the first talk.
Hello, everybody. My name is Corri Desaulniers. I am a 46-year-old mom to 2 teenage boys and a wife to an amazing husband. And we can, next slide. My story began in the fall of 2021 when I had several visits to the emergency room with gallbladder pain. I was set for a gallbladder removal surgery, and pre-scans discovered a mass in my liver. A biopsy determined that it was what they thought at the time a neuroendocrine tumor, and I was scheduled for both gallbladder removal and a liver resection in May of 2022. The surgery was considered successful, clean margins, and nothing else was found at the time. So I was sent home with adjuvant therapy, which I did for 6 months, but no follow-up was completed in that time period. So in August of 2023, is that right? No, sorry, wrong time. April of 2023, we determined that it had metastasized to my peritoneal lining. And at that time, we went through the gamut of all the recommended chemotherapies. I did about a year of gemcitabine, cisplatin, and DERV. We're just going to go DERV because I can't say it. There was progression. We transitioned to FOLFOX, and then came probably one of the second worst days of my life when I was told in August of 2024 that there were no more treatment options available to me within Canada. There were no trials available. There was no drug options. There was nothing. And that's where everything shifted for me. So we can move on to the next slide. So at my lowest point, an unofficial mentor, one of my dear friends, and a fellow cholangio patient encouraged me to reach out to C3. And through that, I was encouraged to advocate for myself, and I pushed. And I was able to get my oncologist to agree for a second biopsy of my peritoneal mets. So, sorry, a biopsy of just my peritoneal mets, but my second biopsy of this cancer. And that through C3's molecular testing program, it was discovered that I had the FGFR2 mutation. And so in December of 2024, I was granted exceptional access to pemigatinib, and as it sits right now, February 2026, I have shrinking tumors and in some cases, and otherwise everything else is stable. There has been no growth in over a year. So we would consider that an amazing success story, I would say, going from zero options to life, to hope, to being here for my children. I remember going to my oldest son's 8th grade graduation and sobbing because I did not think I would be there for my youngest son's 8th grade graduation this coming June, and I will be. So, because of that, I have become passionate. It has changed my way that I see myself and my role as a patient. Information matters. Access to treatments matters. Molecular testing matters. And building your community. Had I not reached out to my community, I would not have access to the information. I would not have been led to C3. I would not have been given access to the molecular testing program, and I would not have gained access to pemigatinib. And not everybody knows how to ask for that. So that is what has become a very important part of my advocacy journey, is helping people become advocates for their treatment plan. We could, next slide, Len. So advocacy for me is not about standing on a stage, even though I guess I technically am doing that today. It's about survival, learning, asking questions, and pushing when something doesn't feel right. And it's also about making sure that other patients know that they can do the same. So why advocacy matters to me? Many patients are misdiagnosed or diagnosed late. Cholangiocarcinoma is rare. Treatment options can be limited, but precision medicine is a changing landscape. Sorry. If I hadn't asked, if I hadn't been nudged towards molecular testing, I probably wouldn't be here today, to be frank. I would have been home waiting to die. So advocacy means encouraging patients, empowering them to take a leading role in their care, supporting awareness and fundraising campaigns, Light It Green, our cholangio cycle, sharing real patient experiences. I will talk to anybody about my story, whether it be, and this actually did happen, a random stranger on an airplane whose father just died of cholangiocarcinoma, because sharing your story helps other patients. And building community, making sure that absolutely nobody feels alone. And so today, advocacy is about, it's part of how I'm coping. It's me learning to live within my new normal. I can't do all of the things that I used to be able to do, and accepting that and working within my new normal has done wonders for my mental health. So if my story leads to one person asking about molecular testing, if it helps bring one family to find hope, if it brings one more person into this community, then it's been worth it to me. So I guess that's really my story, and I hope that I'm sorry. I hope that I can help other people find a clearer path than I had and feel less lost than I did through this journey. Advocacy is not about being brave. It's about making sure that other people are not as lost as I was. And so thank you for listening to me. Lots of love to all of you.
Thank you so much, Corri, for starting off this webinar with such a moving experience. And yeah, you've definitely given me a lot of hope, and I really love working with you, and so happy that you're doing well on this treatment. You definitely are impacting so many people. So we are here with you, and yeah, you're not alone as well. So we're gonna pass that on now to Dr. Robert Grant, and he's gonna talk to us now about the importance of biomarker testing in biliary tract cancer.
Thank you, Leonard. And thank you to C3 for the opportunity to speak today. And thank you, Corri. I mean, it's a beautiful way you shared your story, and I think an inspiration to everybody, and really, I think, sets up what's a little kind of a technical aspect part of your story, and that's biomarker testing, but a really important part of your story. And I'm so glad that you're doing so well. Let's go to the next slide, Leonard. Please. Just click through once. So the great news about, we have a lot of work to do as a community in biliary tract cancer, cholangiocarcinoma, but there's been a really encouraging acceleration when we look at treatments over the past recent years. So for a decade, we really only had one chemotherapy regimen, and then there were some additional chemotherapy regimens, and then immune therapy has come on the scene and is making a big impact. But then there's really been this explosion of what are called targeted treatments. So there's several that are targeting specific to biliary tract cancer, but also treatments that target specific genetic alterations or biomarkers, which we'll talk about in detail, across all types of cancers. And if we click to the next one, Leonard, and these are targets that are enriched in biliary cancer. So you can see just from this timeline the major advances, many of which are in this area of targeted treatment, which target specific biomarkers, and hopefully this trend continues. And so that's the focus of this discussion, which is biomarker testing. One of the challenges, and we talked about this as a group before the meeting, and actually Corri brought up, there's all sorts of different names for this. And that's one of the challenges as we try to help people navigate this journey. Just click through twice, Leonard. Sorry, I didn't know I wouldn't be doing this. So I think the broadest term for this is what is biomarker. So what biomarker is something that we're measuring as an indicator of something related to in this setting, cancer. And biomarkers can do many different jobs. They can help us with diagnosing a cancer, understanding its potential behavior, monitoring. But really the most important biomarker, and the one that C3 and others are focusing on because of its impact, are what are called predictive biomarkers. So tests that we can do to help select the best treatment for patients. And that's what I'll focus on. Biomarkers, other terms you may hear that reflect the same idea are molecular profiling, next generation sequencing, comprehensive genomic profiling, but these are all variations of what I like to call biomarker testing. Next slide, please. So as Leonard alluded, Dr. Vogel, who you'll hear from next, has led the ESMO guidelines, international guidelines on how we should manage cholangiocarcinoma and biliary tract cancers. And this is a flow chart of how you can approach cases. These are guidelines, so everybody's different. But a few things I'd like to point out. The first is that for individuals that don't have the earliest stages of cholangiocarcinoma, molecular profiling is right at the top of the algorithm. Really important. If you click one more time, and that's because these targeted treatments like the pemigatinib that Corri alluded to are available to an important subset of individuals if we identify these predictive biomarkers. And Dr. Vogel's been updating this guideline regularly, in large part because there's been so many new targets identified and new treatment developed in relatively rapid period. And you can see here, they've listed 8 that rise to the level of evidence for the guidelines. Dr. Vogel will talk about clinical trials where many additional targets are being explored. And there are others that are of moderate quality evidence as well. So the message here is that molecular profiling is really important and comes early in the journey for somebody with locally advanced or advanced cholangiocarcinoma. And the goal is to identify what's becoming a very wide array of different potential biomarkers, which I'll talk about, to guide treatments, optimized treatments. Next slide, please. So maybe just click through a whole bunch here, Leonard, sorry. So in biology, like in basic biology, there's this idea of the central dogma. And I think this is important for understanding biomarkers. You know, we inherit our DNA. It's a code, 4 molecules, A, C, T, and G, 6 billion in a string that makes us who we are. And makes us different from each other and different from other animals and plants. What cancers are often driven by relatively small changes in that long genetic code in a bile duct cell. And then that goes on and becomes a cancer. So this code gets mutated. There's this messenger molecule called RNA, which goes then and helps create proteins. And proteins are what actually are doing the work, the function, the structure. And so there's this central dogma of DNA to RNA to protein. And the reason why I like biomarker as a word, or molecular profiling is good too, is it can capture this entire spectrum. So, you know, much of what we're doing is DNA focused, but there are important biomarkers for cholangiocarcinoma focused on RNA and protein as well. Next slide, please. So I've used NGS in this slide. So next generation sequencing, comprehensive genomic profiling is often talking about DNA sequencing. And what that typically is, for example, the C3 panel, which is fantastic and has changed many people's journey, is targeting the key cancer genes that we know can have mutations that guide targeted treatments. There's many more. That's sequencing a tiny fraction of our DNA. The next broader category of sequencing is called exome sequencing. That's all of the regions that sequence for proteins. So thinking back to DNA, RNA, protein. And then you can sequence the entire genome as well. The value of having broad NGS panels or genome or exome sequencing is that these gene lists update over time. And for those who have specific panels, it's important that their oncologist or that they talk to their oncologist, look and say, are there any new targets that weren't on the panel when we did it before? And that can be a reason to retest. So that's DNA sequencing. Next slide, please. One of the challenges is this is a cancer where it's often a real challenge to get enough tissue to properly do any of this testing. It's buried deep within the body. There's often risk to go in to do it. And one of the key things is to try to get large core biopsies, so to get big enough samples to do this sort of genomic analysis. So that's one challenge. The other challenge is that often, and this is a biological property, the cancer cells make up a relatively small proportion of the biopsy, and that's called cellularity. And so what we do in our research program is what's called laser capture microdissection. So we point a laser towards where the cancer cells are and then cut those out. And that's a way of taking what often will be 30, 40% of the specimen is cancer cells to enriching it up to 80 or 90%. But this isn't something that's available commercially or widely. It's very resource intensive. But just to say these samples, obtaining the samples can be a challenge. One way around this, if we go to the next slide, Leonard. This is just showing the benefit of laser capture microdissection. So this technique that we do, if you get higher quality samples, you find more of these targetable. Go to the next slide. For those who don't have targetable biomarkers, and there's concern about low cellularity or enough specimen, another option which can be highly effective at identifying these DNA changes is doing what's called liquid biopsy. So testing a blood specimen, because small amounts of the cancer DNA are released into the bloodstream and they can be tested. And this is a recent study showing that tissue biopsy and circulating tumor DNA can be complementary. The vast majority of the times they agree. Testing that, if you had to pick only one, if you get a good tissue sample, that's probably preferable because you're testing right at the source. But sometimes, probably for these technical reasons, we find things in plasma, in blood, that we don't find in the tumor. So in an ideal world, we might do both of these tests. Next slide. RNA. So this is really important. Corri shared her story with an FGFR2 fusion. This is a complex type of genetic change where two genes are fused together. And this is just showing two potential partners, so FGFR2 on the left and BBP on the right. I won't go into the details, but technically sequencing the DNA here is very challenging because often the fusions happen in between regions of the gene, which are repetitive and challenging to sequence. What's happening here is that these genes get fused and then a new RNA is made. And then a new protein is made. And so the most sensitive way to detect these fusions is not sequencing DNA, but RNA. And you can also see is it overexpressed and driving the cancer. So specifically for FGFR2 and other fusions, we do prefer RNA as the sequencing test. Next slide. And then finally, proteins becoming more important. So HER2 is a really important biomarker, that, just like in breast cancer, an important biomarker in cholangiocarcinomas. And this is a protein test where you stain cancer cells using a stain. You can see here on the top right, a cancer that stains really highly for this HER2 protein. And this can be important for guiding treatment as well. Next slide. Next slide. So as what you're probably seeing is that this is very complicated and technical and specialized. And so C3 has done a fantastic job of bringing the national community together to identify the specialists and bring people together to have conversations like this. What should we be testing? How should we be testing it? And also running multidisciplinary tumor boards. So we have ones within our Legrelli program, but C3 runs fantastic multidisciplinary tumor boards where we review people's stories, but also the specific genetic testing as well. Next slide. And so just to give you an overview of our registry, It's the idea is that it's a real collaboration with patients. Many people want to contribute to science and advancing treatment for the future, for people diagnosed in the future. But the idea is that we also, through the research program, provide this genomic profiling, and we do whole genome and transcriptome sequencing. So all of the and the laser capture microdissection. So all of the technology I've described. And then we review every person's story at our multidisciplinary tumor board. And I thought I'd just share two stories just as an example. You've already heard one from Corri, which is a great circumstance from C3. So this is our research report. So we look at all of the detailed genomic data in depth. We make sure if we look at the top left here that the sample quality is adequate. We look at all of the different mutations, the signatures. But the biomarker that really stands out from this genomic report is a high tumor mutational burden. So this means this cancer has many, many, many more, probably 20-fold more mutations than the typical cancer. And this is a biomarker for immune therapy. And this was an individual who, for other clinical reasons, may not have done immune therapy, had a contraindication, an autoimmune condition. And because of this report, we were very optimistic about immune therapy. It shifted the balance of potential harm and benefit. And we tried immune therapy, and it was really remarkable. Had induced a major shrinkage that was durable. So that's one example. Another patient, if we go to the next slide. So same information, quality, signatures, different mutations. This cancer had a mutation in a gene called BRCA2. So this is a gene you may have heard of in the context of breast and ovarian cancer. People can inherit pathogenic variants. We used to call them mutations, and that can increase people's risk of cancer. This is different. It's the same gene, but it's mutated only in the cancer cells. And with the genome sequencing, you can look and see that it has all of these downstream consequences. It changes the whole genomic landscape of the cancer. And makes platinum-based chemotherapy more likely to be effective, which it was in this circumstance, and also opens up the possibility to have maintenance treatment with pills, a targeted pill called olaparib. And that's what this individual, that's what the journey has been so far. So just to go to the last slide before I conclude. So we've just about quarter of the way through our journey with this program. We reported our initial results. This is an open access study anybody can read. Now, half of our participants, half of the people contributing, had specific genomic alterations at our tumor board that could be targeted later in their journey. Among those who had gone enough in their journey to the second line treatment to receive it, half of them did receive targeted therapies. But the challenge at the time was that none of them were available through the public system. So that's changed in large part because of the work of people here today in advocacy, like pemigatinib, for example. But this was the early experience and really encouraging, and other tumor boards around the country have shown similar benefits. So hopefully that was helpful. Thank you, and I'm looking forward to Art talking about clinical trials.
Thank you, Dr. Grant. So Dr. Vogel is going to share his screen, and then he's going to present on an update on clinical trials in Canada, and I'll let you take it away.
Yeah, thank you very much for inviting me, and I will give you an update on clinical trials we have at the moment here in Canada, and it I think nicely builds on what Robert just has mentioned before. He has already shown you the ESMO clinical practice guideline, which we have updated last year. And I want to start with the early cancer. Here in the guideline, we have recommended adjuvant treatment. So after resection, we would recommend to add additional chemotherapy for 6 months with an oral drug called capecitabine. The efficacy, I mean, it has shown efficacy in clinical trials, but we are not really happy, and we would like to improve outcome after resection. We know that patients are still at risk of recurrence even after extensive surgery. And there was one trial which actually proved that it's really feasible to do clinical trials in the adjuvant setting. So this is a study from AstraZeneca, which actually has finished recruitment already ahead of schedule. And here a new bispecific antibody has been tested as an add-on to chemotherapy, we were able to include a couple of patients in this trial, and we are very much looking forward to this treatment. So this would add, if this trial is positive, immunotherapy to adjuvant chemotherapy. Many of us, however, think that maybe it's not only important to add chemo and immunotherapy after resection, but also before resection. And at the moment, we are conducting a clinical trial here in Canada. At the moment, only the site here in Toronto at PMH is open, where patients can be included if they have an intrahepatic cholangiocarcinoma, are fit and fit all the in and exclusion criteria, and then they would get chemotherapy, 2 cycles of a triplet chemotherapy plus this bispecific antibody, the immunotherapy that has been tested in the adjuvant setting. Then after surgery, when patients have recovered, they will proceed to adjuvant treatment, which is a little bit more than what we are doing at the moment. It's only capecitabine, as I just mentioned, but here we would do this chemo doublet, gem-cis, what we also do in the palliative setting, and also add the bispecific antibody as it has been done in the adjuvant trial. So, we are following here a little bit the strategy to what we are doing in other GI cancers at the moment, that we are not doing adjuvant treatment, so chemotherapy after surgery, but adding systemic treatment before surgery. And I think that's a very good option for our patients we can offer at the moment. For patients that have more advanced disease and are no candidates for resection, Robert already mentioned the molecular profiling, the molecular tumor board, which is definitely very important. Our standard of care at the moment is gem-cis, the doublet chemotherapy, plus a checkpoint inhibitor, which could either be durvalumab or pembrolizumab. The trials were positive, have shown that patients live longer if they receive this checkpoint inhibitor. So this is today's standard of care. And luckily, we do have more and more patients that really survive for 2, 3, 4 years with this really devastating cancer. However, we definitely can do better, and we still would like to improve outcome for our patients when they receive systemic treatment in more advanced stage. And again, here, coming back to this antibody, rilvegostomig, which is this bispecific antibody, double checkpoint inhibitor, if you like, which is combined with our current standard of care, gem-cis, and compared to what we would do otherwise as standard of care, gem-cis and durvalumab, which is one of the tested checkpoint inhibitors I have just shown you. So you can see at the moment this bispecific antibody, rilvegostomig, is pretty much in the center of ongoing efforts to improve outcome, both in the adjuvant setting. The study has been completed in the neoadjuvant setting. We have just started our trial, and this study also is ongoing, combining doublet chemotherapy with this dual checkpoint inhibitor. So I think it's also a very good and interesting option for our patient. Then, as highlighted before, we have all these targeted therapies for patients with cholangiocarcinoma. And here, as Dr. Grant has highlighted, molecular testing is really of utmost importance. We do have a couple of drugs and combinations that are approved by FDA, by European agencies, and which are also available here in Canada and approved here. So there are definitely options. Not everything is reimbursed already, but there are always ways and which we could try to get even drugs and combinations that are not yet fully reimbursed. Yeah, so it's definitely important to do the testing, and then we can explore how we could get the treatment. And there are a lot of targets, and this is a little bit what has been shown before. Many different targets, which therefore require broad molecular testing, which means that we do not only look at one single gene, but multiple genes. And this is important when you ask and reach out to your oncologist, that the right test is really employed for this molecular testing. Yeah, because many times I still do see patients coming to me for a second opinion, and then I get the note, oh, there was nothing in my panel. And I always say, nothing is impossible. There always needs to be something. And if there's really nothing, then we need to relook at the test again and maybe redo the testing again, because some of these alterations are really very difficult to detect. So rebiopsy, as also mentioned before, can be very important. I think I don't need to go into all these details. We have seen a couple of approvals, and the question is now, how can we go forward and how can we do better? And there are, I think, two strategies. One is that we are moving these targeted therapies into the first line, and this is important, and probably the best strategy to overcome primary and secondary resistance to these treatments. So the idea is, when we combine the best we have, which means chemo, immuno, and targeted therapy, we can really get the best possible outcome. And there are actually a couple of trials which are ongoing at the moment. You have heard before about FGFR2 fusions and pemigatinib, which was the first drug that was improved. I was pretty much involved in this clinical trial, which led to the approval of pemigatinib. And you can see there are a couple of drugs which are now available to treat these patients with FGFR2 fusion. Importantly, however, there are also new drugs, and these new drugs can be employed in patients that have been pre-treated with first generation FGFR inhibitors. So pemigatinib is now available in Canada. We have another trial where we can use futibatinib as an FGFR inhibitor. But eventually patients may progress, which of course may take a while. And now we have new drugs like tingotinib, which is effective in patients that are progressing on pemigatinib. And there's actually a phase 3 study, unfortunately not running in Canada, because it is still so difficult to get FGFR inhibitors in the first place here. So the company decided not to open trial sites here in Canada, but we do have a very close by trial sites in Buffalo or in Chicago. And I have sent a couple of patients to these sites to be included into the first 308 study. And one patient is now for more than 6-7 months, I think, on treatment with tingotinib after progression on pemigatinib. So I really think it's a very, very interesting option for patients that have progressive disease on pemigatinib. HER2 has been mentioned before. It's great to see that we have now approval for the treatment of BTC with zanidatamab. Here, the multiplex immunohistochemistry is very important. So not only NGS testing needs to be done. These biomarker testing can be really confusing because different methods and tests are employed. So one is the genetic part, where we do broad molecular sequencing, and the other part is the immunohistochemistry, which we also use to basically characterize these tumors by histology. So multiplex immunohistochemistry is very important, and I think all patients with GI cancers should be tested for HER2. And this is important that it's done as early as possible, because we do have a clinical trial in the first-line setting for patients with HER2 positive tumors. We have seen very interesting data in gastric cancer at ASCO GI earlier this year. This was a study also led by one of the medical oncologists from Princess Margaret Cancer Center. And the very similar trial design here in cholangiocarcinomas, where we basically combine GemCis doublet chemotherapy with a checkpoint inhibitor, immunotherapy, and the HER2 targeted therapy, in this case, zanidatamab. And I think this is a very, very good option. But it requires that testing is done as early as possible, because it's a first-line trial, and patients need to have basically the test results in hand before they start systemic therapy. So I think these are the trials I wanted to mention. We do have trials more or less in all stages of the disease. Neoadjuvant, adjuvant has been just finished recruitment. A couple of first-line trials also for IDH1 mutations. So again, testing is important, because we have now at least 2, I think 3 first-line trials for patients with druggable alterations. So doing this testing as early as possible is important. And then we do have these trials in second line. And as I've shown you for FGFR2 fusions, we can apply FGFR inhibition as sequential treatment. So patients can not only be treated with one FGFR inhibitor, but also with the second FGFR inhibitor, which definitely offers more opportunity now and in the future.
Thank you for that, Dr. Vogel. Yeah, I think that from the last two talks, it really seems like we're kind of scratching the surface. Dr. Grant showed that from the sequencing that we're doing now, the biomarker testing, it's really just a portion of all that we can look at that for possible therapies. And also now Dr. Vogel shared just some of the clinical trials that are available in Canada. So I think that there's still so much more to come, and there's still a lot to be hopeful for. I'm going to pass it off now to Dr. Rebecca Auer, and she's going to share a little bit about the C3 and then also lead the panel discussion.
So thank you so much, Leonard in particular, for doing a lot of the lion's share work in organizing this, and Rob and Arndt for agreeing to speak. Thomas has been an incredible supporter with Servier of a lot of the work we've done with C3 and for supporting this webinar. And I just especially wanted to shout out and thank Corri for sharing her story. I am the caregiver for my husband, who is a cholangiocarcinoma patient diagnosed in 2022, May 2022. And I wanted just to tell Corri how much it resonated with me, because it was just a few months before my eldest, I have 3 teen boys as well, my eldest grade 8 graduation. And I also cried thinking that it would be unlikely that my husband would be there for other graduations. And I'm really excited to say my eldest son is going to be graduating from high school at the end in the spring. And so I think there's so many reasons to hope, and just wanted to share that I completely understand your story. So thank you for sharing. So I'm just going to talk a little bit about C3. After my husband was diagnosed, I really felt that there was so much promise when I spoke to people like Rob and Arndt and other experts in the field, but I didn't feel like that hope and that sort of excitement that research was bringing was filtering down to Canadians who were being diagnosed with this rare and devastating disease. And so together we worked on this application to receive funding through the Canadian Cancer Society and CIHR to be able to create a collaborative that was really designed to be a partnership between patients, researchers, clinicians, caregivers, to really help to foster hope through research. And we have 3 pillars. I lead the hope pillar, which is really the patient connection pillar, molecular testing rounds. Talk about that. Dr. Simon Turcotte is leading the STAR pillar, which is a clinical trial looking at infiltrating T-cell therapy for cholangiocarcinoma. And certainly some challenges in getting that therapy to patients, but we're very optimistic that we'll get that trial up and running very soon. And then Dr. Ilko, who is a PhD scientist I work with, incredibly brilliant, and she has a team across Canada looking at novel therapies in the preclinical pipeline that I hope will be available to patients. As I mentioned, C3 is really a partnership. It's about growing hope together through access to research, molecular testing, expert opinions, and a supportive community. Leonard and team have really done an incredible job getting our name out there to the community. We're being very intentional about building this community. So we have, of course, our website, cholangio.ca, and patients can sign up, and we send all of our patients a care kit with a handwritten note, a message of hope from either a patient or a researcher, caregiver, oncologist. We also have a Facebook group. We organize local meetups and have had one sort of 1.5, but national meetups called Meet C3, where we get patients and researchers and oncologists all together. We have webinars and C3 chats, as well as a book club, support groups, advocacy groups, and offer scholarships for patients to attend meetings and conferences to again help bring information back to the patient community. We also have a strong network of medical experts, mostly medical oncologists, but also surgical oncologists, pathologists, radiologists, interventional radiologists, radiation oncologists, all of whom have an interest or expertise in treating cholangiocarcinoma. And we connect with them through national multidisciplinary rounds, which we hold every month and discuss complex cases. We provide them pamphlets to give out to their patients, to let their patients know a bit about C3 and what we do. We engage with a lot of them, and Rob and Arndt and many physicians at Princess Margaret have been very vocal in advocacy and trying to advocate for both pemigatinib most recently, but then molecular testing for patients. We have a physician newsletter where we also provide information about the latest both trials and or recent developments in cholangio research. As I mentioned, this is the STAR pillar that Dr. Simone Turcotte runs. Essentially, the concept is you can take the immune cells out of a patient's tumor. So you have to resect a portion of the tumor, you remove the immune cells, you culture them, and you select the ones that are attacking their own cancer. So you have the patient's own cancer that you kind of use to see which ones are reactive. And we can also screen them to see whether they're reactive to certain abnormal proteins within the cancer. And then we expand them to sort of 1 billion cells and give them back to the patient to fight their cancer. And so this has had some success in the US, predominantly at the NIH. We're excited to be able to offer this to Canadian patients, hopefully in the near future. And then this is the impact pipeline therapeutics. So we have researchers all across Canada, from BC right to Dalhousie, working on different novel therapeutics to bring to hopefully to the future for patients with this disease. So a few program highlights. We do have a registry, so we want to collect as much data as possible from patients. And our key principles are that we think patients should have the right to manage, access, and share their own data. But sometimes patients are not really able to navigate the system. And so we want them to be able to delegate data collection and uploading to us so that we can help them facilitate having their charts moved to different providers. And we think that patients should be able to participate in research as part of the standard of care. And sometimes getting into a clinical trial requires that you have all your records. And so this is hopefully to help facilitate them, their participation. We also think a national registry will help us determine if patients are eligible for clinical trial, especially if a new clinical trial opens up. It'll give us some real world evidence to really hopefully make the case of how important molecular testing is. And then we've been working with the International Cholangiocarcinoma Patient Registry so that we can share data with them and have our data as part of that, so we can really understand from a much bigger picture the cholangiocarcinoma landscape, prognosis, outcomes, etc. We already have over 400 patients enrolled in the registry since it opened. We did have a molecular testing program that was funded. I'm very sad to say we've had to close that as of the end of December 2025, but in 18 months, we were able to offer over 280 patients molecular testing, and you can see it was from across the country. You'll note that Quebec isn't there because Quebec has been able to provide molecular testing funded by the provincial government, and we had a number of clinicians participate across the country. And our goal now is to figure out how to advocate for this to be funded by the provinces as part of standard of care, because ultimately we have been able to see how beneficial this can be. I mean, Corri's story is an excellent example. Dr. Grant provided a couple of examples. My husband is another example of patients who are alive and doing well with relatively good quality of life because they had access to molecular testing and a targeted agent. In our testing, we've estimated about 40 to 50% have potentially actionable genomic mutation. That means that there is a drug available either off-label or as part of clinical trial today that could potentially target that mutation or that biomarker in that cancer. So we think this is the real reason why we absolutely need to insist that every patient gets molecular testing. And it really allows us to expand the treatment options beyond our standard of care. And of course, every one of our patients in the molecular testing program is also in the registry, so helping us to really understand that full landscape of patients. As I said, unfortunately, the program did have to close, but we are certainly going to be using this data that we've been able to collect and continue to collect to provide advocacy, to get molecular testing, to help patients still get information about clinical trials. And we still can support patients in getting molecular testing. We just don't, unfortunately, have any additional funds to pay for that molecular testing. And I know someone said in the chat, how do I interpret the results of my molecular testing? So certainly you can talk to your own physician, but that's also what C3 is there for. So we're happy to connect with you and help to interpret your molecular testing for you in language that makes sense and see what opportunities there may be based on those results. So we're continuing to work on the registry, ensure that we get full data collection for all the patients. It takes a bit of time to procure all the records, so we're still working on that, but the real goal is to generate real-world evidence, particularly on the benefits of molecular testing. But we really need to advocate for molecular testing to be part of a standard approach in every province, not just Quebec. And so we really need you to be involved and help push this forward. If you're interested, we have an advocacy group that we run. If you have ideas or suggestions or how we can move this forward. But I think the next step for C3 is really a very strong advocacy campaign directed to both federal and provincial levels of government to ensure that patients with this rare cancer have access to the treatments that could change their journey completely through availability of molecular testing. We do have a petition that is open at change.org, and we'd love for you to join and tell your friends to join our petition, really advocating for funded molecular testing across the country. So that's the end of my part. And I think we could maybe just launch right into the panel discussion. And Corri, I don't know if you want to put your camera back on. So
Yeah.
again, thank you everyone for joining. Leonard, did you want to jump in before?
No, just going to ask all the speakers to unmute and then turn on their videos so I can spotlight them.
I think this first question, if I'm not mistaken, was for Dr. Grant. Is that right? So what challenges do you see with tissue availability for biliary tract cancer, and how does that impact the testing success? And I think you touched on this briefly as well.
Yeah, I think part of it is education, just like what we're doing here, like getting the word out to the patient, but also really the medical community. Often this is a cancer diagnosed on what are called brushing. So you just get a few cancer cells under a microscope, and you have enough for the diagnosis, but you don't have enough for this biomarker testing. So I'm getting the word out to everybody that getting, as much a big a specimen as safely possible, say through a core biopsy, is something that can be actually done for many people. The other is liquid biopsy. I mean, we're advocating for tumor testing, but I think looking at where this field is going, having access, particularly for those where we don't find an alteration or there's concerns about adequacy of the tumor specimen for biologic reasons like cellularity, having access to liquid biopsy would be very important as well. So I think that it's a fundamental challenge of these cancers, pancreas, biliary, all in the bile ducts here. But there are strategies to overcome it, and we just need to look forward to doing that.
And is that, if a patient has a brushing and they get the diagnosis, let's say they go on to therapy, initial response, but then progress, like Corri did, and have a more solid nodule, let's say it's in the liver, in the peritoneal cavity, in the lung, at that point, should they be advocating for another biopsy because there could be actual tissue for molecular testing. Like that's always a bit of a challenge, but I think we should be advocating for that second test if the first test is not adequate.
I agree. And everybody's situation is different. We have to make sure it's safe, but I agree. Targeted therapy is so important after first line. It's really important. And it might even be earlier in the journey. Like if you just have brushings, I think it's reasonable to go back and try to get full genomic profiling in many cases.
Okay. The second question is for you, Corri. And you touched on a bit as well, but maybe you could just outline what information or support do you wish you had earlier when navigating molecular testing and clinical trial access?
Oh, so much. But I think Dr. Grant already commented on, first off, the language around biomarker testing, genomic profiling, molecular testing, molecular profiling. There's so much terminology that is used interchangeably that it can be confusing. And for quite some time, my oncologist thought I was asking for genetic testing, not biomarker testing, not molecular profiling. And so I didn't know I had even had testing done on my liver. So, I mean, it's quite confusing. So being able to have an educated conversation with your oncologist. So I guess knowing the right things to ask your oncologist or your care team in general would be a big thing. Also knowing the importance of comprehensive molecular testing, and getting all of the information out there. Because when I was initially treated, I was given little to no hope. So, if I had been connected with C3 sooner, connected with the cholangio community, I would have had a lot more hope and a lot more empowerment to guide my treatment journey. So I guess that would be it in a nutshell.
Thanks, Corri. I think your comment about comprehensive biomarking or molecular testing is so important because when we think about the different, for example, Health Canada approved treatments, pemigatinib requires like an basically an RNA-based molecular test. The IDH1 ivosidenib, that requires like a DNA-based test. And then the newer HER2 therapies, that requires something called immunohistochemistry, which isn't like a sort of traditional, I guess, next generation sequencing or genetic or genomic test. Although it is, but it's a totally different format. And so these words are really complicated if you don't know much about molecular or biomarker testing. C3 is here to support you in that as well. So we can help you have that conversation with your oncologist too, because I know some patients have said to me, well, my oncologist said I got testing. Well, what did they test for? What testing did they get? And they don't know because they didn't know to ask that, right? They just wanted to know, did I get biomarker testing? And that can mean a lot of different things. So really clarifying what kind of testing did you get? What important negatives as well as positives came up? Did they test for this? Was it negative? Did they test for this or not? Those are really important questions, and we shouldn't be feeling badly about asking them. Even if your oncologist doesn't know the answer right away, they can find out for you. I think it's really important. Thank you, Corri. Our last question, this one is for Dr. Vogel. What gives you hope about the future of precision medicine for biliary tract cancer in Canada and over the next 5 years? And actually, I'll open this one up to Corri and Rob as well after.
So I think there are two things. One is that we are getting more and more access not only to targeted therapies but also to the testing. So when I came to Canada 3 years ago and we had our initial discussions, I think it was even more difficult than it is today. So I think also specifically thanks to C3, we have really made a lot of progress, and this is promising. It's reimbursement is still an issue here. Yeah, but we are making progress, and we have now more and more access to these drugs. And this is good, right? I mean, we should not be left behind what is offered in other countries, and I think we need to make awareness for that. Altogether, physicians, patients, and I think if drugs and combinations are approved by like global leading regulatory agencies in the US and in Europe, I think we should have the trust, and also the regulators here in Canada, they should have trust that these are effective treatment, and then they should be offered to our patients. Yeah, I think we definitely need to. I mean, we made progress, but we definitely need to keep this pressure up. Because there's still a lot of drugs which are not easily available here in Canada, and we need to continue on that path. Having said that, since we have now more and more access to testing, we are testing for FGFR2 fusions now, for example, will offer more opportunities in the future. So I think we will not have the situation that a company decides against Canada, like we have seen it for the tinnigotinib trial, and that these companies are also coming to Canada, which offers us also opportunities to treat our patients with these drugs. And then lastly, I think at the moment, the pharmaceutical companies, but also academia, are making so much progress in understanding the biology of these tumors, that I'm really hopeful that even the undruggable alterations, so alterations for which we do not yet have therapies available, will become eventually druggable. And I mean, KRAS is one example. It's very frequently mutated in many cancers, specifically in cholangiocarcinomas, but also pancreatic cancer. And we thought for many years that we will never have drugs for these genetic alterations. And now we see more and more RAS inhibitors which are employed. So that's very hopeful. And I think with these efforts, we will make progress, and I think it will be faster than in the past. So I mean, it's still a very difficult to treat tumor, but I'm very hopeful that we will make considerable progress in the near future.
That's great. Thanks. Dr. Grant, did you want to add anything to that?
No, that was exactly all the thoughts I was going to say. I think, thankfully we've seen a lot of new drugs coming, but thanks to Dr. Vogel and others' efforts, there's a lot of really exciting new drugs on the near horizon. Dr. Auer, everything you've been doing to bring the national community together has been really important for Canadians. So I think there's a lot of work to do, but the future is bright, and we've made a lot of progress in the past few years, and I think it's a trend that should continue.
Fantastic. And Corri, last word to you.
I think for me, Dr. Vogel sums it up with the word progress. That gives me hope. So if we can keep that momentum moving forward, there's just that's hope for everybody.
Thank you, Corri. I'll hand it over to Leonard. I'm not sure if we have any additional time for questions or.
Yeah, we have some time. I think we'll go until 1:45. So there are some questions in the chat. Maybe Rebecca, do you mind taking a look and asking them? I'm gonna just get the slides up again.
Great. When an NGS test is run on a tissue, would you be able to capture different fusions and mutations in one chest, like FGFR2 fusions and IDH1 mutations? So, Arndt, why don't you field that one?
Yeah, I mean, that's exactly the case. I mean, when we do, I mean, we can do tissue testing and blood testing. Blood testing, I think, is an alternative when we do not have enough tissue. I think it's not yet as perfect as tissue testing, because the tests, I mean, they're improving, but they're not yet as reliable as tissue testing. So blood testing is only like an alternative if you do not get enough tissue. But I think in most patients, and Rob has also discussed that, I think we are able to get sufficient tissue. It requires education and some efforts, and sometimes we need to do 2 or even 3 biopsies. But in most patients, we can get it. Then it's important that we are using the right test, right? And this is, I mean, something I think we should not ask our patients to discussed this with our colleagues. I mean, this is something what we, I think, have discussed many, many times in the last 2, 3 years. And I think now everybody is very much aware of the difficulties of testing. And I think we are now at a point where I'm not so concerned anymore that the wrong test may be employed, but it still may happen. So again, what I have highlighted in my slide a little bit, if there's nothing, you should always be concerned because there always must be something. And if somebody tells you there's nothing, then be concerned and ask for a second opinion.
Yeah, I think it was a really good point that by and large, I think when C3 started, there was a lot of times when patients would get a biomarker test and it wouldn't be adequate for FGFR2. I think now
Exactly.
that's much improved.
Yeah.
Although I would still say that immunohistochemistry for HER2 amplification is not being done routinely and maybe something
No.
that needs to be asked. So
Yeah.
yeah.
And I agree. And maybe to come back to this, the problem is there are no clear guidelines yet. And I mean, there are many pathologists which are still a little bit hesitant because they say there are no clear guidelines for HER2 testing in cholangiocarcinoma. I think that's a not valid concern. I think the concern may be valid, but it still should be done. And I think we have a clear recommendation that the guidelines for gastric cancer should be followed. But this is something that still requires efforts, and patients need to ask it. And it's so important because it's just an immunotherapy, which is much easier and faster than broad molecular testing. And we have these trial in the first line setting. So asking for that is really, really important. And I understand there are still problems, but this is definitely also something we need to work on. So HER2 testing needs to be done. I think in all GI cancers, I mean, colon cancer, gastric cancer, there's no cancer in which we do not have no benefit for HER2 targeted therapy. So it should be reflex testing as MSI. There's no reason not to do it.
No, that's a very good point. I have
Yeah.
a comment here from Heather Blake. Excited to see some of my mutations mentioned P10, P53, FGFR4, but I have no idea how to take advantage of this. So any advice if because those are, I think, certainly not mainstream to have targets.
I think we need the MTD molecular tumor board for that, right? And we have C3, we have our tumor board in at PMH, and the field is so rapidly evolving, and it's very difficult to keep track on everything. So there's only one recommendation, and which means you need to go to a molecular tumor board where it's discussed by experts. Otherwise, it's impossible to keep track.
Yeah, so that's a really good point. So if you would like your case discussed at our molecular C3 tumor board, the best is if your physician can request it because it's easier for us to get the files. However, you can also request it yourself if you're part of the registry. We can procure your files and connect with your oncologist. Particularly if you're looking for a second or third line option and you have some molecular results and you're not sure how to interpret them. I have from W. Anderson, why are patients not told about these testing options even if they are not covered by provincial coverage? I know that can be a hard one because it's hard, especially in our Canadian system. It's very hard to offer patients something that it can be quite expensive. I mean, it can be upwards of $4,000 to get the molecular or the biomarker testing. And so it's always a bit tricky to tell a patient that you could get this, but if you don't have the money, you can't get it. And I think it's just a challenge. I think we are becoming more comfortable as a medical community in bringing it up and letting patients decide. But I think it's hard for us. The biggest change, what we want to see is that we don't have to have these challenges where people who are wealthy enough can get it and people who aren't don't get it. We don't believe in that in Canada, and it shouldn't be the case for this rare cancer. So we keep working on that.
Yeah, I was just going to add, I think a lot of these challenges and questions that we've talked about, having like a centralized, provincially funded program would lead to standardization, awareness, Like there would just be so many knock-on benefits. Leonard's put the what do we call it? Petition in the chat, and I think we all need to work together to make this possible, because many of these challenges I think would be knock-on benefits of having this access.
It's time to get loud, is what I say. It's time to get loud and annoying so
Yeah.
that finally they listen.
Yeah. Yeah, so I think that's a great place to end the meeting. Again, there is this petition that we started, and I will clarify that is for Ontario specifically. Right now, the reasons are Ontario's largest population or province with the population-wise in Canada, and it is provincially funded. So we think, start with Ontario and the others will follow. There's also an email there too. You can email info@cholangio.ca if you want to share your stories. If you've had molecular testing done, or a loved one's had it and it's led to, being put on a trial or getting access to a drug, we want to hear those stories so that we can share them and have more, I guess, information and things that we can use to advocate for it. One more announcement slide. So this Thursday, February 19th, is World Cholangiocarcinoma Day. And every year on World Cholangiocarcinoma Day, there's lighted green events, and essentially landmarks across Canada are going to be green, and also across the world. And it's to stand in solidarity with the patients and the families affected by cholangiocarcinoma, and just to show some support. So there's a QR code again on the screen, or you can go to mychcc.ca/WCD. On that website, you'll see a list of all of the locations that are going to be green, and some of them even have a local meetup happening. And so I encourage you to reach out to the contact information there if you are in one of those cities. And if you do happen to go to one of those events, please take a picture of yourself with the landmark and send it to us, and then we can share that too. It's all about raising awareness for this rare cancer. And I think that, yes, we are definitely seeing some progress being made, but there's still so much more to do. But we're here to do it alongside you. So just want to thank again all of the speakers. I want to thank Survey as well for co-hosting this and putting this together with us. And we hope to see you again next year, and also before next year's webinar as well. So with that, we'll close the meeting, and it'll also be posted online soon. So thanks, everyone.
Thank you.
Thank you. Bye-bye.
Bye.
Thank you, everyone.
Thank you. Bye-bye.